By the age of 65, 10% of people are expected to develop Alzheimer's disease. By the age of 85, that will be half the people in this room right now. Alzheimer's disease is the most common form of dementia, characterized by severe memory and cognitive function deficits. While there are therapeutics available right now, none of them successfully reverse these effects. Biologically, Alzheimer's disease is caused by the accumulation of plaque in the brain. Microglia, the resident immune cells of the central nervous system, help clear these plaques in the beginning stages of the disease. However, in late-stage Alzheimer's, these microglia become dysfunctional and can help progress the disease further. One hallmark of these dysfunctional microglia is fat or lipid accumulation. But how do the microglia become dysfunctional to begin with? Interestingly, while the brain is the softest organ in the human body, these plaques are as stiff as glass. This brings me to piezo-1, a mechanosensor that can detect these changes in tissue stiffness. I aim to link piezo-1 activity with dysfunctional microglia fat accumulation. My project uses both mouse and human microglia with and without piezo-1. These cells are dosed with various concentrations of yoda-1, which is an activator for piezo-1, and are subjected to different substrate stiffnesses. These cells are then analyzed using both microscopy and Western blotting. As I mentioned before, one hallmark of dysfunctional microglia is fat accumulation. But is this accumulation attributed to uptake or biosynthesis? I investigated CD36, which is a known regulator of lipid uptake in microglia. This Western blot shows its expression level with an increasing dose of yoda-1. The band intensity correlates to the amount of CD36 present in the sample. I found that an increase in yoda-1 leads to a subsequent decrease in CD36 expression. However, this is not so noticeable in the piezo-1 knockdown, which indicates that the presence of piezo-1 decreases lipid uptake, which is the first time that this has ever been shown experimentally. The functional results of this data can be seen using microscopy, in which the fat appears in pink. When yoda-1 is added to the cells with the presence of piezo-1, there is a significant decrease in lipid uptake. Interestingly, piezo-1 can be engineered to bind to specific tags, and when combined with Tor microscopy, I can visualize it specifically on the cell surface. And as we can see on the right, piezo-1 is in real time. While there is still so much research to be done in this field, our findings do indicate that piezo-1 activity plays a significant role in microglia lipid biosynthesis. These findings can further be investigated to develop novel therapeutic targets for piezo-1, delve into the differences between mouse and human models, as well as create a better understanding of the causality and possible treatments for Alzheimer's disease. And with that, I'd like to thank the members of my lab and our collaborators, and thank you so much for listening.